Hello everyone. Today I'm going to present our work titled Neurological Status Classification using Convolutional Neural Network. So my talk will be roughly divided into four sections. Firstly, in introduction part, I'm going to talk about our motivation and problem definition. Then I will provide some information about the dataset and pre-processing steps, as well as some background knowledge about artificial neural networks, followed by our proposed model. Then I will show the performance evaluation of our model and finally a summary and conclusion for this work. In conditions such as diabetes or seizures, a deterioration in patients' health condition has been reported to be linked to a surge in stress level. To manage and control such occurrences, we need to be able to track the neurological status. This is usually possible by studying the neurological signals such as electroencephalography or EEG, electrocardiography or ECOG, and functional magnetic resonance imaging or fMRI. However, these methods require clinical conditions and in some cases like ECOG are invasive data collection methods, so they have uncomfortable and time-consuming protocols. On the other hand, given the rapid growth in development of minimally invasive sensor built-in devices, as well as prevalence of using wearable devices, it is now possible to collect great amount of different types of physiological signals, some of which have been proven to have relevance to the changes in neurological status. So, given this information, we want to design a system which can use these non-invasive physiological signals to track and study the neurological stages. Artificial deep neural networks are powerful tools which can be used for multi-class classification problems when enough data is in hand. Here in Figure 1, you can see an overview of a system where a dataset is comprised of various time series, is fed to an artificial deep neural network, and then after a nonlinear transformation, the desired output is obtained. So our objective is using this concept to propose a deep neural network to discriminate different neurological stages given a multivariate dataset including various physiological time series. For this purpose, we used a publicly available dataset collected with response sensors from 20 college students undergoing an experiment with the purpose to probe a subject's response to different types of stress. Here you can see the experiment design for data collection. In this experiment, there is 5 minutes recording for each of the four stages: relaxation, physical activity, cognitive stress, and emotional stress. Relaxation is considered as the baseline while physical activity is broken into three parts with different intensities: standing for 1 minute, walking for 2 minutes, and then jogging for 2 minutes. After another 5 minutes relaxation, the individuals experience cognitive stress stimulation consisting in counting backward a list of numbers for 3 minutes and then taking the Stroop test for 2 minutes, which is reading the name of colors written in different coloring and saying what color the ink is. Finally, after another 5 minutes relaxation, subjects are shown a horror movie for 5 minutes to be emotionally stimulated. In order to avoid redundancy among all the relaxation phases, we only included the first one into our computations. Moreover, the first 40 seconds of cognitive phases during which the subjects received explanations on the clinical protocol regarding the experiment were considered as emotional phase given that subjects showed patterns similar to emotional patterns. Moreover, here in Figure 3, you can see an overview of the data collection protocol. Using the pre-mentioned experiment, subjects experience different stress levels, as you can see here, meaning various levels of arousal. At this stage, physiological signals are recorded using two response sensors, one recording EDA, accelerometer, and body temperature at 8 Hz, and the other one recording heart rate and an estimate of amount of oxygen in the blood calculated by oxygen saturation called SpO2 with sampling rate of 1 Hz. Here you can see a segment of the recorded signals. So we take this dataset and extract the variables that have been proven to be useful in studying the neurological status. Firstly, we aggregate the 3D accelerometer signal into one-dimensional signal by taking the square root of the summation of all dimensions, which is equal to the magnitude of the 3D accelerometer. Here, L represents the length of signal. For SpO2 and heart rate, we smoothen them by filtering the noise out. Here in figure 5, you can see these 3 physiological variables for patient 1. The other variable that we use is the electrodermal activity or EDA. There are many studies that have shown that features extracted from EDA signal in both time and frequency domain are relevant for neurological status analysis. So we take the EDA into consideration as well. However, EDA can be modeled as a sum of 3 terms: the phasic component, which is an impulse-shaped signal describing the sudden changes in EDA signal, the tonic component, which is a smooth-shaped sequence corresponding with the slow changes in EDA, plus an additive noise. Here in figure 4, phasic and tonic components for patient 1 is depicted in the left hand and right hand side respectively. So to summarize, the physiological variables that we are going to use for training our models are these 5 variables: accelerometer or ACC, heart rate or HR, oxygen saturation or SpO2, tonic component or T, and phasic component or P. From previous study of our group, we have found that there are relations between changes in neurological status and frequency component of simultaneously recorded physiological signals. So for this purpose, we take the absolute magnitude of the short-time Fourier transform or STFT for each of the 5 variables to capture their frequency content over time. Here in this equation, xt is the signal content in time domain, w is the function, capital X is the Fourier transform, and tau and omega are time and frequency indices respectively. Here in figure 6, you can see the spectrogram showing the changes in the magnitude of frequency content of the ACC signal for patient 1 over time for 4 different neurological stages, which are relaxation, physical activity, cognitive, and emotional stress. In this color bar, moving from yellow to dark blue color corresponds to moving from the maximum to minimum spectral density respectively. It should be noted that for each window of length 30 timestamps, we take the frequency content that has the maximum magnitude. We slide this window all the way to the end of the dataset and for each variable separately. By this, we obtain 5 more arrays of information corresponding to the frequency component of each physiological signal that we had previously. Moreover, we take the first, second, and third order time derivatives of all variables and append the resulted 15 arrays to what we had previously, resulting in 25 arrays of variables. Since we are dealing with a multivariable dataset now, to solve the problem of having various distribution for each variable, we apply a z-score standardization method on each of the variables separately. Here in this equation, xi is the observation, mu I and sigma I are the mean and standard deviation respectively. After this, we concatenate the data for all subjects lengthwise, and finally, we use a window of width 25, the same as the number of our variables, and the length 30 with 50% overlap, and slide it all the way to the end of the dataset to create samples or segments of size 30 by 25. So we had a dataset of size 144,000, which is the length of the dataset, and we turn it into 14,000, almost 14,000 samples, each sized 30 by 25. We do this to make the dataset ready to be fed to our model. Before talking about our model, I will provide some required background knowledge about the neural networks. Generally, artificial neural networks, or ANNs, are categorized as a type of machine learning algorithms inspired by the human brain, consisting of a stack of neurons called layers, such that the neurons within the same layer are not connected to each other. Here in Figure 7, a simple neural network with one hidden layer is shown. When a network is properly trained, the optimal value for weights, which are shown as W's here, are obtained such that given a set of observation or input, the model can predict the corresponding output with the least error with respect to the target or desired output. The more layers a model has, the more complicated problem it can solve. So given this fact, we introduce deep neural networks, or DNNs, which are types of ANNs with multiple in-between layers or hidden layers connecting input and output layers. In a DNN, each layer receives the output of the previous layer as the input, applies a nonlinear activation function, and passes the result to the next layer. Then in the output layer, we get the predicted values by the model, which based on the type of the problem may have different types of hyperparameters and different numbers of neurons. So in this type of model, we can have as many layers as we want, but it should be noted that the deeper a model is, the more data it requires to be trained properly. Moreover, we can have different model architectures based on the type of problem that we are solving. So convolutional neural networks, or CNNs, are well-known deep learning architectures with specifically excellent performance in computer vision problems. The most important benefit of them is automatic feature extraction, which makes them a very powerful tool when dealing with a multivariate dataset. This model is made up of blocks of convolution and pooling layers followed by one or more fully connected layers, which are the typical neural networks that we showed in two previous slides. Basically, we have three types of CNNs: one-dimensional, two-dimensional, and three-dimensional. The difference between them is one-dimensional CNN is able to derive features from fixed length slices of a dataset by using one-dimensional convolution filters. This property makes 1D CNN a very powerful and fast method in identifying simple patterns within a data, which can be used for more complex patterns in higher layers of the model. However, in 2D and 3D CNN, two and three-dimensional kernels are used respectively, so they have advantage in solving more complex problems while they are computationally more expensive than the 1D CNN. It should be noted that 3D CNNs are mostly used when we have video datasets due to the capability of extracting features from both spatially and temporally, which is not our case here. Here in Figure 9, you can see how a 2D convolutional layer performs. A kernel slides all over the input, which is usually an image, or in the other words, a tensor or 2D sensor, and performs the convolution and maps the resulted value to a new feature map at the output of the convolution layer. So given this background knowledge, we introduce our proposed model. We use CNN models for the purpose of multivariable time series classification. So we propose two models, a 1D and 2D CNN model, which receive our physiological data and by layering the patterns in it, classifies the input into four neurological stages: relaxation, physical activity, cognitive, and emotional stress. So here in this figure, you can see a representation of both 1D and 2D CNNs because, as I just mentioned, the difference is in the kernels that are not shown here specifically. So in our model, the input layer receives the segment of size 30 by 25 from dataset that we have already prepared. Next, in this layer, or in the convolution part of the model, using three blocks of convolution and pooling layers, significant features and informations are extracted, and then the resulted feature maps are flattened and fed into a fully connected layer where all neurons have full connection to all activations in the previous layer. And finally, at the output layer, we have 4 neurons representing 4 neurological stages. Then, neurons with the maximum value means the maximum likelihood that the input segment belongs to that class. So after training the model using different hyperparameters, the best results obtained by using exponential linear unit or ELU, tangent hyperbolic or tanh, and softmax as the activation functions of convolutional, fully connected, and output layers respectively. Here you can see the equations for these three functions. Plus, we had root mean square propagation or RMS probe as the optimization algorithm with learning rate of 0.001 to minimize the loss function, which is categorical cross entropy in our model. We split the data into 70% for training, 10% for validation, and 20% for test. We trained 1D CNN and 2D CNN for 100 and 150 epochs respectively with batch size of 30. Here in Table 1, you can see the performance evaluation of each of the proposed models. Results show the precision, recall, and F1 score for detecting each class separately, as well as the average accuracy of the model in classifying 4 stages. You can see that both models show great performance while the 1D CNN shows slightly a better accuracy. It should be noted that due to having less trainable parameters, 1D CNN is significantly faster than 2D CNN, so it can be a better choice in this case. Moreover, we compare our proposed model with 2 of the most well-known classifiers, support vector machine or SVM and random forest or RF. Given the results in this table, we can see the superior performance of the CNN model, the proposed models, over these 2 well-known classifiers. In multi-class classification problems, confusion matrix is very important because it shows how the model is confused in discriminating different classes. Here in figure 11, we see the confusion matrices for 1D CNN on the left-hand side and 2D CNN on the right-hand side. It is observable that both had very small confusion in discriminating cognitive stress from emotional stress, which is probably because the physical activity or the accelerometer signal is almost the same in those 2 stages, but we can see that this confusion is very few. Finally, to challenge our proposed approach, we added a white noise with zero mean and standard deviation 1 to all of the sequences and trained all previously mentioned models again. Table 3 shows the performance of all methods resulted after training with the noisy dataset. Again, it is observable that CNN model significantly outperforms SVM and RF in presence of noise, this time even with a greater difference. However, it should be noted that in this case, 2D CNN outperforms 1D CNN, which is because of having 2D kernels, i.e., 2D CNN has more trainable parameters and can learn more detailed features from the dataset. Hence, it can be more powerful than 1D CNN in more complex problems. So to summarize, in this paper, we exploited a deep learning approach to solve a neurological status classification problem using a multimodal physiological time series dataset recorded from wrist-worn sensors. We obtained 99.82% and 99.61% classification accuracy, as well as 99.78 and 99.77% F1 score for 1D and 2D CNN models, respectively. Our proposed model outperforms 2 well-known machine learning classifiers, which are SVM and random forest. The proposed model shows superior performance in presence of noise, which is a very important criteria while working with datasets which are prone to be distorted like physiological datasets.